perhaps I'll just kick off by introducing myself and the company very broadly. So my background is I did my PhD in plant microbe interaction. So I studied how the communities of bacteria and fungi in the leaves are affected by various different synthetic chemical pesticides and also plant defense hormones. And after completing my PhD, I moved into industry. So I worked at another London-based company as an environmental microbiologist. And then I joined Concert Bio just 2.5 years ago now. So I lead our biological research and development team here. So my team cover the microbiological, horticultural, and molecular biology aspects of the business. In terms of us as Concert Bio, so we moved into our first lab here in West London in 2022. So we're still a very new company and still a very small company. So we're a team of 12 overall. And our overall mission is to develop the first biostimulant product targeted for hydroponically grown crops. So terrestrial crops have been co-evolving with very rich soil microbiomes for a very long time. And that very rich soil microbiome provides a lot of benefits to the host plants. I think functions that people might be most familiar with are fixing nitrogen or solubilizing phosphate. So biologically available forms of these nutrients are in very, very limited supply in the soil. So the plants need to rely on the microorganisms that colonize them to carry out these functions for them. And the soil microbiome provides other benefits as well. So it's able to activate plant immunity by producing plant defense hormones, and also helps the plants directly protect against pathogens by inhibiting or competitively excluding the growth of pathogens that come from the soil as well. The challenge we have here in hydroponic systems, which I'm sure you guys are very, very familiar with in your team, is that the plants are removed from this very rich reservoir of microbes. And in hydroponic systems, they often use these very inert substrates that have very, very attenuated microbial activity. And there's a trade-off here, right? Because on the one hand, the good thing about using these types of substrates is that the chances of a disease being introduced to the system in the first place are relatively low. However, what it does mean is once a disease gets in, it's likely to be even more devastating in terms of the outbreak and the consequences to the system, because these plants just don't have this protective microbiome and very rich microbiome like they do in soil. And the people who really feel these consequences are the growers that are using these methods. So they employ these methods because they want improved year-round crop productivity, particularly with the challenges like climate change that we're facing. But they're still, as a consequence of this depleted microbiome, seeing reductions to yield and very devastating consequences of disease outbreaks. So there is really a need to restore the function of the microbiome in these CEA or hydroponic systems. There are some existing solutions out there. So probably the most commonly implemented one is traditional synthetic chemical pesticides, such as fungicides, which can control fungal or fungal-like diseases such as Pythium or Phytophthora. But these chemicals have undesired environmental effects. I think they're discussed very widely in the press. And as a consequence, they're increasingly not being renewed for registration. So the choices of them out there that are available to growers are becoming smaller, and the adoption of them is becoming lower as well. And as a consequence, growers are increasingly turning to biological solutions to address these challenges. Not only do they want to avoid the harmful environmental effects as well, but it's in keeping with their ethos, because these people are always very close to the earth. However, the ones that are out there that exist commercially at the moment are really very broadly designed for traditional soil-based agriculture. And although some of them do provide instructions for hydroponic growers, they're sort of retrofitted, and hydroponic growers are more of an afterthought than they are their initial target market. And this can create a lot of challenges for hydroponic growers in terms of implementing them. So one of the first problems that growers encounter is that some of these existing biocontrol microbes that are available in the market, they just simply don't survive in hydroponic systems. The two species that you guys might be familiar with are Bacillus and Trichoderma. They're very commonly used biocontrol microbes that are used for the control of things like Pythium or Phytophthora. So this is some data that we've gathered from hydroponic microbiomes, and you can see the Trichoderma and Bacillus there in the green, and this is their relative abundance. So this is the percentage of the microbiome that they occupy. And you can see here that actually it's much, much lower than the disease Pythium here. So we've actually seen even worse data than this, and we see growers that actually really try and put very large quantities of them in their systems, and we can't even detect them at all. So let alone them surviving in levels that are sufficient to outcompete something like Pythium, which it can even reach something like 10% of the abundance of the total microbiome during a severe outbreak. Secondly, we just simply don't really know what mechanisms are beneficial in hydroponic growing. So in traditional soil-based agriculture, there's always this challenge of limited biologically available nutrients, but that just doesn't translate as well really to these systems. So these plants are grown in a solution that is nutritionally optimized, and these roots spend their entire life cycle completely stooped in biologically available nitrogen and phosphorus. They don't need that microbial intervention in the same way that soil-based plants do. And the actual physical environment is very different. So nutrient solution is not like soil. It lacks a physical matrix, and it's aquatic, and the temperature is different. So for a variety of reasons, not only can most soil microbes not survive in it, but they also just may simply experience suboptimal growth. We've also done a lot of work with growers over the years and just find that the efficacy in general is low. So many of the growers that we work with have trialed these things that are on the market for years and years and just really have never been very convinced by the success or by what they've seen. In fact, one of the things that we hear the most is that they once introduced something like a trichoderma, but because it's not harming their system, they're now scared to withdraw it. So in fact, even their use might be based on something like less than empirical data. So with those challenges in mind, the company was established with the mission of trying to discover beneficial microbes that are tailored specifically for hydroponically grown plants, rather than ones that are more retrofitted from soil-based agriculture. And the way that we do this, it's broken into 3 parts. So firstly, we've been collecting microbiome data from hydroponic facilities all over the world, and we've been combining that data with yield and environmental data that growers provide to us. And from that, we can build machine learning models that help us see which of these microbes are putatively beneficial. So by using the environmental data, not only can we also correlate changes of different microbes with changes in yield, so this microbe goes up and the yield goes up, but we can also account for any environmental variation that might have explained why the plant yielding increase. So for example, a greenhouse grower, their plants might simply be growing better because it's warmer, and it'd be as simple as that. So by using all of this yield and environmental data, we can hopefully really try and pinpoint the effect down to a particular microbe. And once we've kind of got that list of microbes that we think are beneficial, we'll apply some further selection criteria and design some experiments to test them. So we can test them both in our in-house facilities, and we've also tested them and in commercial trials as well. One of the things that's quite useful about the approach that we use is we can also take samples from our trials as well and feed these back in. So every trial we have, even if it's a failure, we learn something from the data every single time, and we only improve the models that we build. So the process of collecting our samples, we call this our monitoring service, and we think that we've now got the largest collection of hydroponic microbiome samples collected from real-world systems in the world. And it's a service that initially, when we were developing products and first building our candidate list, we provided as a standalone service to growers, but we still continue to do so because it's actually of an added benefit to them as well as us. So in terms of our monitoring service, we took our first samples in 2022, and we've now reached more than 18,000 samples that we have from hydroponic facilities all over the world. So this is 57 different greenhouses and vertical farms that we've taken samples from. We initially were also focusing in terms of the crop that we were targeting. We were focusing on leafy greens in the beginning, particularly lettuce, but we've since been expanding into various different crop types over the years. And we also collect data on the type of system that they're using. So we have samples from nutrient film technique, deep water culture systems, ebb and flow systems, aeroponic systems, and drip irrigation systems. And by taking all this data and combining it together, we've now identified more than 40,000 unique bacterial and fungal or oomycete species from these hydroponic systems. So they're not quite as absent of microbes as we think, but still compared to soil, that's still relatively poor in terms of the diversity. So we haven't just taken them from the UK, so different species of microbes, they're dispersed geographically as well. So we really have tried to cast a fairly wide net. So a lot of the samples that we've collected have come from North America and continental Europe, and even some in the Middle East as well. Actually, this graphic is now a little bit out of date, and we've got some from the Caribbean since, I think, too. So we're also casting that net in terms of the diversity that you would see in different pockets of the world as well. So in terms of how we do this, we like to think that the monitoring service that we provide is end-to-end. So we send kits out to growers. So if they want to take root samples, we send tubes that are like this, and they put their roots inside there, and these tubes are filled with a lysis buffer. So it will lyse the cells and it will preserve the DNA. And if they want to take samples of the nutrient solution, we send them syringes and we send them filters like this, and they'll pass their nutrient solution through the syringe and through the filter, and they just send the filter back to us. So it actually just captures the DNA. And the idea behind this is if we're getting sent samples from all over the world, we want it to be as user-friendly as possible and not have our users subject to using cold chains and things. So it's a way of them being able to send samples to us where they don't really have to think about it too much. They can just put them in a box and they'll stay perfectly stable for long periods of time in transit, and various issues with customs as well, although we're pretty good at that now. So once they reach our lab here in West London, we extract the DNA from them. And then in terms of the technology we use third generation amplicon metagenomic sequencing. So some of you guys might be familiar with next generation Illumina sequencing. We use one called Oxford Nanopore technology sequencing. And because it's third generation, it simply means that the actual length of DNA that we get is longer. So it just gives us a bit more confidence when we're trying to say, okay, well, that bit of DNA is this microbe. And then from that, we have a downstream pipeline that we use. So one of the early challenges we faced is that because there aren't loads of people collecting lots and lots of hydroponic data, we actually found that a lot of the out-of-the-box software and workflows and databases even, they just weren't up to scratch for the needs that we had. They either didn't cover all of the microbes that we wanted to cover, or they were built with Illumina or next generation sequencing in mind. So we built a proprietary in-house bioinformatics workflow to give them the best assignments that we possibly could. So what that workflow does is it takes all these many reads of DNA and hopefully turns it into something human readable, which is ultimately a long list of microbes and figures to say, okay, we've detected this microbe and we've found it this many times as well. And that's where that data is suitable to go into our machine learning pipeline. So we can feed that microbiome data in and pair it with the yield and the environmental data that the growers provide to us, and hopefully use that to try and discover beneficial microbial candidates. So one of the things that we found was that not only are we interested in this data, but we found that the growers themselves who we were taking the samples from were really interested in this data too. So we created a dashboard for them to be able to access the samples that they've sent to us and the data that we produce from it. So we can also help to educate them on the microbiome as well. Now, this data isn't always the simplest to grasp. Microbiome data is by nature very complex with lots of weird and wonderful Latin names. So we tried to make something that was as palatable for them as possible. They, unlike us, they don't have the fortune of being able to sit around all day and look at microbiome data. And there are a few metrics that we're able to show them. So one of those that we can show them is their diversity, so the number of species that we were able to detect in their system. Another one that's very important, you can see it in the middle of the first panel there, is the ratio of pathogens to beneficial microbes. So what we found increasingly is that it's actually not unusual at all for a grower who isn't seeing any disease symptoms to still be experiencing some low level of disease pressure, and that in and of itself isn't necessarily a concern. Where it becomes a concern is actually if there aren't many microbes around it that are not pathogens to be able to sort of control it and to keep it in check. So that's another piece of data that we can provide them is the kind of a health index of their microbiome. And one thing our dashboard allows them to do is to track these various metrics over very long periods of time. So, for example, a greenhouse grower might experience disease challenges more generally in the summer than at any other time. So they might not want to say, okay, what are my disease levels like this month versus last month? They might want to say, okay, what were my disease levels like in summer 2025 and how do they compare in summer 2024? And can I correlate that with a change in my yield as well? So speaking of disease monitoring, it's still by far the biggest use case that we have of the dashboard, and certainly the things that the grower seems to be the most interested in, and why they're very motivated to send us samples. The growers that are concerned with plant diseases and that have experienced diseases in their systems, they tend to be very concerned with the detection of diseases like Pythium and like Phytophthora, sometimes Fusarium as well. And one of the things that's very nice about the approach that we take is because the sequencing method that we use is agnostic, it means that even if we can't say specifically which Pythium species it is, we can still tell them that there is an unidentified species of Pythium in their system. And it's one of the benefits it has over something like, for example, qPCR, where you'll have an assay developed that detects very specific things. We're at least able to say, okay, we're not sure what species it is, but there might still be something going on here. And there's also another group of growers that are very interested in human diseases that have they have very, very stringent food safety protocols. So these tend to be more the vertical farms. And of course, their primary motivation is that they really don't want to make somebody unwell at all. But also if there is a disease detected in their system, it can cause these very big halts in production. So they're very interested in the dynamics of where and when in their system do they find human diseases, and if there are any environmental things that they can do or any interventions that they can carry out to minimize that. So once we've utilized all of that microbiome data and put it through our data science workflow, we sort of end it in the overall workflow here in the second segment. So we've started with these whole microbiome data sets. And from there, correlated it with yields to produce a list of microbes that we've identified as being potentially beneficial. But there is some further narrowing that we do as well. So ultimately, we want to make a product for people, and that does mean that we want something that's going to be able to scale in terms of its growth. Something that we're always very concerned with is actually only a very small subset, about 1% or so, of known microbes are suitable for growth in lab environments. So we also narrow that down based on that. Anything that we think, okay, maybe this won't scale very well in production. And then after that, we're ready to test them in either commercial systems or in-house, and hopefully develop products from them. So when we started out collecting these microbiome samples, I mentioned that we were largely targeting the leafy green market. And part of that motivation was that leafy greens have a very short life cycle. So it means that while we can see success fast, it means we can also fail fast and learn quickly as well. So we have since developed our first product, Overture, for leafy greens. So you'll have to forgive me while I give the obligatory shameless sales pitch for a second. But us, particularly in the R&D team, have also started to expand into new crops as well. So in 2024, we sent the top 2 performing microbes that we had from these initial trials out to as many people as we possibly could, and named them Overture A and Overture B. And over the course of the year, we ran 14 trials in commercial systems. We did some trials with independent contract research organizations, and also had some help from a couple of different universities across the world. And from that, we were able to obtain a 9.9% average improvement in yield. And normally when we spoke to growers, they've said that 5% moves the needle for them. So we were very happy with that result. And that constitutes an 80% win rate. So in 82% of the trials that we ran, we had some success. So of course, it's not completely perfect. It's not 100% win rate. And there were some trials where we weren't able to see a yield improvement, some of which we think might be down to these very many complicated combinations of different substrates, different varieties, and different system types. So someone might have variety A and they're growing it in the nutrient film technique system and using quire, and perhaps it just doesn't work with that particular combination. However, we do find that when we've sent both to growers, we've really never not been able to see an improvement if they've been able to trial both. And interestingly, some of them tell us that one of them works a lot better than the other. We're still really trying to learn and to find out why, but we'll take the win. So in terms of what Overture is, so it's a microbial suspension concentrate, so it's in liquid form. And the video here of a grower applying it with their spray bar, so it's being sprayed directly onto freshly sown seeds here. And this is probably the easiest way for them to apply it. Obviously, we can't get them to install big expensive pieces of equipment to be able to apply our product, so we have to think about their processes as well when we do product development and when we designed Overture. And this is very nice and simple for them. And we do always try and apply it to the seed, particularly in leafy greens, and that's due to a concept called the priority effect, whereby the earlier a microbe can arrive and has a chance to colonize, the more likely it is to be successful in that colonization. So we really try to encourage them as close to apply it as close to the sowing event as possible. And so here's Paul, who's our CEO, in a silly red hat. Sorry, Paul, if you're still listening. And here's him with the operations manager from Jepco. So Jepco are the largest hydroponic lettuce producer in the UK. And this was our first commercial sale of Overture, which occurred back in November in 2024. And one thing that's very nice and very full circle is that Jepco were the first grower to send us samples for our monitoring workflow. So it was very nice to be able to make them our first customer as well. And just a little bit more in terms of the next steps for our leafy green product is we ultimately want to launch into the US. So in the US, our overture will be called Prelude, and there's a glimpse of the product label there. We've now got regulatory approval in several states, so including Texas, Georgia, and Massachusetts. Those are the ones there on the graphic in green. And we aim to have approval for the states there in blue by the end of the year. We'd also like to have regulatory approval in the EU by the end of the year as well. But whilst the commercial team are very heavily focused on launching that product into the US, us and the R&D team have started to pivot into other crops as well. So we've recently run our first cucumber trial, and we saw a 10.7% improvement with the numbers of cucumbers harvested with one of the microbes. And with another microbe, we saw a 5.65% improvement in yield. And actually, this was quite surprising for us because I think the expectations around how biostimulants will perform in vine crops tends to be a bit lower. I think for growers that grow vine crops, they consider something like 2% a very large increase in yield, as just gains in general tend to be a bit more incremental than they do in leafy greens for a variety of reasons. So this was a very nice result for us. And it was also a fair bit of a learning curve as well, because even to learn about how yield is recorded in vine crops compared to leafy greens, it tends to be relatively straightforward in leafy greens. You chop the head off the lettuce and weigh it, and that's as far as it goes. But these, for example, these are called snack cucumbers. So they are actually picked to spec. So if they are too small, they simply won't be picked, so they won't be included in the harvest. Hopefully they'll be included in the next one. And the grower doesn't necessarily get paid any more or less for larger fruits. They have to be within a size spec, and that's it. So we're really, for cucumbers and snack cucumbers like this one, really trying to target an increase in the number of cucumbers produced, because otherwise the grower simply won't get paid anymore. So another thing that we've found very interesting is that vine crops can also be steered between two different phases of growth. So they have vegetative and generative growth. So they can be steered between either producing fruit, which is the generative stage, or having enough leaf biomass, which is the vegetative stage, to produce the sugars that they need to produce the yield in fruit. So this was something that we actually saw with our recent first tomato trial. So this was carried out by an independent contract research organisation here in the UK. And we were very excited that of the 4 microbes that they were able to trial for us, one was able to provide a 15.2% uplift quite consistently. But when we looked at the breakdown of the harvest data, this is something very interesting as well. So they did 2 rounds of harvesting. They had a first and a second harvest. And if you look there at candidate 4 in the purple, you can see an enormous increase in yield to the point where we couldn't really believe it in the first harvest. And then it massively falls off in the second harvest. So what we think is actually it was really driving the plant towards generative growth in the first harvest. And it meant that there wasn't enough vegetative growth by the second stage. So it sort of almost exhausted itself early on. But we thought this was very interesting, both to show not only what these microbes can do, but we're also very pleased to have our first tomato result as well. So just to round off, these are our teams. I mentioned we're a very small team of 12. I'd like to thank particularly Paul, our CEO, for being the machine that keeps everything running. And also my colleague Phoebe, because she actually does most of the production of these microbes. And my colleague Maurice Barrett, who actually designed our proprietary bioinformatics workflow that enabled us to discover these microbes in the first place. And we also have my colleague Diamond, who actually runs our monitoring pipeline. So she's the person processing all of the samples and putting them on the sequencer. And we've also got a number of really wonderful collaborators from all over the world that have helped us to get this data and to make this work possible and help us to launch our first product. So I'd be very remiss to not thank them as well. So, and thank you all for listening, and very happy to take any questions that you have.